So it's talking about the shortening of your DNA, especially on the lagging strand side of the DNA replication. Yeah, because that primal length of DNA is left uncopied, so that much will be shortened every time. Okay, then you will see a problem if your cell goes through so many replication processes. Then its chromosome will be shortened, and then what if there are important functional genes located there? You lose that gene, then that will be detrimental for the cell, right? That is a possibility, but normally at the tip of the linear chromosomal DNA, especially in the eukaryotic cells, the end of the chromosomal DNA has repetitive sequences instead of functional genes. Repetitive sequences are found. We call those repetitive sequences as telomeres. Telo means like the end. Meres, the repetitive sequences found on the end of the linear chromosomal DNA. So actually the telomere sequence, if there are multiple telomeres there, the telomere sequence will be shortened every time cell goes through cell division, because the replication will take place, and replication will shorten the end of the chromosomal DNA. So yes, the telomere length will be shortened as a cell is going through replication, as a cell is aging, because it has gone through so many cell divisions. So telomere length could be a sign of cell aging. And then if the telomere length is shortened significantly, and normally that kind of signals the cell is too old to carry on its life, so it may go through a cell death. It's we call it the self-cell suicide. Apoptosis is what the terminology is. So cell death is normally initiated if the telomere length is too shortened. So that telomere length is very important for the cells to sustain its life. However, if your cell is specialized in dividing all the time, just like stem cells, okay, or adult stem cells, or the embryonic stem cells, and cells that are dividing all the time, even the cancer cells, okay, then how would they deal with the shortening of the telomeres? Okay, it turns out those cells will have a special enzyme. So this is the telomere repeat, TAGGG. So you have so many of that repeats at the end of your chromosomal DNA, and the length will be shortened in the normal cells. But if your cell is specialized in cell division, it's dividing all the time, then it should have a way of dealing with the telomere shortening, right? Yes, the answer is yes, and it has a special enzyme complex. Okay, the enzyme is made out of a reverse transcription enzyme. So it's a reverse transcriptase. Also within the complex, there's an RNA template coexisting with the enzyme. So there's that telomere enzyme, telomerase enzyme. Actually, the name of the enzyme is telomerase. That's a reverse transcriptase enzyme. Also, it carries the RNA that can function as a template DNA. So you can make DNA off of the RNA template. So that's why it's called reverse transcriptase enzyme. So the entire complex is called so it's a telomere and repeat, that's RNA template, and it has telomere and reverse transcriptase. So it's a complex including the RNA template plus reverse transcriptase enzyme, this TRT. So these guys can see the DNA off of RNA template. Okay, so it can recreate telomere length over and over. So see, using the RNA template, you just make DNA, so that's a reverse transcription that is managed by this enzyme, reverse transcriptase. Okay, so you can go over and over. It can, make lengthy telomeres. Okay, so telomere length can be recovered actually by the third complex, telomere and reverse transcriptase, and then TR, telomere and repetitive sequences. Okay, so actually it can be recovered. Once the telomere end is recovered, so between those two strands, the bottom strand is recovered, right? Then there could be a primer made, and then that can be I guess that can be made again by the DNA polymerase. So both strands can be recovered from cell division. So that's how you can replenish the telomere length in the actively dividing cells like stem cells. Okay, they have the active telomerase enzyme with the RNA template there. That's how they recover the shortening of the telomere length. Okay, so telomere binding proteins are there within that, chromosome end, and then they tend to regulate telomerase activity. Okay, regulate telomere activity and telomere length by inhibiting the telomerase activity. So you don't want to, make telomerase active all the time, right? Then you can go make your chromosome very long by repeating the telomere over and over. So that activity, telomerase activity, is regulated by these proteins called telomere binding proteins. That's what it says. When they are binding fully and telomeres fall off, so there's no more synthesis of telomeres happening. Okay. So telomere binding proteins also protect chromosome end by forming T-loop at the end of chromosome. So instead of exposing the end like this, it forms a loop like this, and it's kind of cross over as a T-loop. So it's called T-loop. So by forming T-loop, this end is not accessible for DNA chewing enzymes. That's how they can protect the end of a chromosome. So that's the structure, and that's the telomere and the telomerase activity. Okay, so that's it for this lecture 5.